Ito ng pag-unlad ay may mga institusyong nagtutuwan sa mga hamon ng panahon.
Picard supports S&T Human Resource Development. Nagsusulong ito ng mga programa na naglalaman ko for better delivery of S&T products and services.
Sinusupport yung mga dalubhasa, sanat, upang umasense
Buluhan ang mga kabuhayan, mamamayan.
Nagbibigay kami ng teknolohiya, nagkasan, at magpapaunlad sa kabuhayan at
may ng pagtawid ng grupo.
Picard pag-unlad. At panglikay
narito UST Picard upang tulungan ng sa sektor maging teknolohiya.
Sa pagsibol ng isang bagong umaga, hatid nito lang lagi ay pag-asa. Makabuluhang pagbabago dito sa sulok ng mundo. Makabagong pamamaraan ang siyang naging daan. Tangkilikin ang likas na karunungan, sa layunin na lahat matulungan. Sama-samang adhikain, nabigyang tuot pansin, ating baya'y pagyamanin. Kung kaya nila, kaya din natin. Kalikasan, kalusugan, kagandahan, kabuhayan at kaunlaran. Pilipino, yakapin ang pagbabago tungo sa katuparan ng pangarap mo. Salubungin ang pangakong bagong bukas, kaalamay gawing sandigan at lakas. Sa pagtuklas ay may biyaya upang lahat ay guminhawa. Ating baya'y pagyamanin, kung kaya nila, kaya din natin. Ito ay para sa kalikasan, kalusugan, kagandahan, kabuhayan at kaunlaran. Pilipino, yakapin ang pagbabago tungo sa katuparan ng pangarap mo. Kaya din natin. Kalikasan, kalusugan, kagandahan, kabuhayan at kaunlaran. Pilipino, yakapin ang pagbabago tungo sa katuparan ng pangarap mo. Sa tulong ng siyensya at teknolohiya, maaabot ang pangarap mo. Yeah, 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 yeah. Yeah, 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 yeah. Yeah, yeah, yeah. Pilipino, yakapin mo, yakapin mo. Pilipino, yakapin ang pagbabago. Biotech na tayo.
Good afternoon to all our live viewers in Facebook Live. You are now watching the 18th National Biotechnology Week, or NBW DOST card webinar on S&T Agri-Aqua Biotech applications, broadcasted at the DOST Facebook page. On behalf of the Philippine Council for Agriculture, Aquatic and Natural Resources Research and Development of the Department of Science and Technology and the organizers, I would like to thank you all for joining us in today's webinar. I am Dani Casimbrano from the Crops Research Division, your host for this afternoon. With the theme Responding to the Challenges, Business Opportunities in Biotechnology, this year's NBW celebration aims to highlight the local biotechnology products and promote the latest biotech innovations that are ready to be transferred or commercialized. As of this moment, we already have 100 viewers from all over the Philippines. Most of our participants are students coming from different state colleges and universities, local colleges and universities, private universities, and secondary schools. Once again, thank you for being with us today, and we hope that you will be able to join us all throughout the entire broadcast. I am sure that all of you are already excited to learn more about these technologies that our research speakers will present today. But before that, I would like to call on the Executive Director of DOST Picard and advocate of biotechnology himself, Dr. Reynaldo V. Ebora.
Pleasant afternoon to all our online viewers tuned in today via Facebook. Council of Agriculture and Natural Development of the USTP on SNT Agri-Aqua. Council annually celebration of the National Technology Week, or NBW. Technology-related research initiatives in collaboration with our partner agencies throughout the country. This is one of the Council's efforts to disseminate and potential contribution and benefits of biotechnology to the agriculture, aquatic, and natural resources, or AANR, community, and economy. The NBW is held in observance of Presidential Proclamation number 44, series of 2007, institutionalized. It is celebrated in recognition of the positive impacts of the safe and responsible use of biotechnology and its products and meaning security, health services, and a sustainable environment. The industry NBW celebrates the team responding to the business opportunities with various government agencies. The technology has active application ability. It has aided farmers in providing available clean planting materials, developing pest and disease-resistant crops, improving agricultural and aquatic management and conservation practices, and increasing the nutritional value of produce. Biotechnology has provided innovations, new strategies, and opportunities to increase the production of economically important agricultural products. For today's forum, we will be seeing 3 of DOST Picard's initiatives on biotechnology that were supported in response to significant challenges in the agriculture and aquatic sectors. Our expert from the National Institute of Molecular Biology and Biotechnology of the University of the Philippines, Los Baños, or Biotech UPLB, will share about the developed microbial-based foliar biofertilizer Trio, which now helps improve the growth and yield of sugarcane plants. Another expert will give us update States on the African swine fever virus nanogold biosensor test kit at Central Luzon State University. Kit is proven to be a cost-effective test for rapid detection and monitoring of the ASF virus, thereby supporting the efforts of the country's industry. Meanwhile, our invited expert from the University of the Visayas will talk about the production and benefit of protein hydrolysates from marine species in loss fish species. This innovation enables the production of alternative food products beneficial especially to immunocompromised consumers. These three biology innovations are only among the many R&D initiatives of the Council that are now potential avenues of business opportunities in the country. We hope that through this webinar, we would cultivate a deeper understanding and applications and benefits of biotechnology. Rest assured that DOST Picard will continue to support SNT emerging technologies, strengthen its partnerships and cooperation among various research institutions, and utilize agricultural biotechnology for a better AANR sector. Thank you and mabuhay tayong lahat.
Thank you so much, Dr. Ebora, for your welcome message and for giving us the rationale of NBW's annual celebration. At this point, we would like to acknowledge our partners, members of the Interagency Committee, and member agencies of the DOST for the collective effort in making this week-long celebration possible. This year's celebration is spearheaded by the Department of Trade and Industry, or DTI. And as mentioned by Dr. Ebora, this year's S&T Agri-Aqua webinar will feature 3 biotech research initiatives in the agriculture and aquatic sectors. But before we get started, let me remind our viewers of some house rules. That first, when posting your questions or comments in the comment section of this FB Live post, please write to whom your question is addressed to. And next, type in your name, affiliation, or location before typing in your question. Also, for those who would like to receive an e-certificate for this webinar, you may accomplish our customer satisfaction feedback form, which can be accessed through the link that will be shown later at the end of this webinar. And now that everything is in order, let us move on to our first presenter. So our first speaker is a retired researcher at the National Institute of Molecular Biology and Biotechnology, or Biotech, of the University of the Philippines, Los Baños. She is a PhD holder in soil science with a minor in biochemistry. She was given the Invention Disclosure Award on December 2018 at the Philippine Genome Center, University of the Philippines, Diliman, Quezon City, for the project development and field testing of endophytic bacterial inoculant as new biofertilizer for improved production of eggplant and sugarcane. As she was also granted licensing agreement in May 2018 by UPLB for the startup on the manufacturing and commercialization of Nutrio. Currently, she is the proprietress and owner of Fulmite Agricultural Corporation, to talk about high yield at lower cost with Nutrio biofertilizer for sugarcane. It is my privilege to introduce to you our first speaker, Dr. Virginia M. Padilla. Ma'am G.
Hi, Dan. Good afternoon to all our FB Live viewers.
Hi, Ma'am G. Ma'am G, can you give us a preview of how Nutrio biofertilizer can help our farmers? I'm sure excited na malaman yung mga viewers natin tungkol dyan.
Okay. Nutrio is a foliar spray biofertilizer that can help our farmers reduce inputs and cost of production, increase crop yield, sustainable, and increase the farmer's income.
Okay. Thank you, Ma'am G, for answering our question. No? I'm sure that our participants are now very eager to learn more about the use and benefits of the Nutrio biofertilizer. So, take it away, Ma'am G.
Okay. With the introduction from our host, Dan, I also would like to acknowledge DOST and PICARD, DTI, and all the organizers of the 18th National Biotechnology Week for this presentation. Please watch my video presentation.
Good afternoon to everybody. My presentation this afternoon is entitled High Yield at Lower Cost with Nutrio Biofertilizer for Sugarcane. The current problems in crop production, particularly sugarcane, are the following: poor growth, prolonged drought, flooding, low soil fertility, high soil acidity, and high cost of inorganic fertilizer, resulting to low national average yield of 60 ton cane per hectare only. One of the solutions to those problems is Nutrio, a bacteria-based foliar spray biofertilizer, promotes nitrogen fixation, produces plant growth promoting hormone, and Nutrio biofertilizer acts as a supplement to inorganic fertilizer. Here are some of the benefits of using Nutrio. It promotes healthier and greener plants, promotes tillering. Based on past field experiments in different locations, increase in yield range from 10 to 30% or even higher. In these two farms indicated in the table, in 1/2 farmers practice plus Nutrio, there is a 50% reduction on inorganic fertilizer with an additional cost for Nutrio, but with a corresponding average increase of 37% in ton cane per hectare and 53% in LKG per hectare, or number of 50 kilogram bag of sugar per hectare. Nutri-foliar spray biofertilizer enables medium-scale sugarcane plantations to improve profitability by achieving higher yields while reducing the use of inorganic fertilizer by 50%, meaning to say less chemical fertilizer, less production cost, increased yield, long-term sustainability, and increase in customer revenue. The development roadmap of Nutri-foliar spray biofertilizer, the commercialization journey. In 2012 to 2018, Nutrio was developed, which was funded by DOST card implemented at Biotech UPLB. We also did toxicological study and pilot testing of Nutrio in regions 3 and 6. And we have also formed the company Fulmite Agricultural Corporation, FAC, as a spin-off company. We were licensed by the University of the Philippines, Los Baños, to manufacture and distribute Nutrio. And on the same year, 2018, we are SEC registered, BIR certified, mayor's permit granted, licensed by FPA to manufacture and distribute Nutrio. And also on 2019, we constructed a small laboratory and purchased equipment for manual operation for a small-scale production of Nutrio. In 2021 to 2022, a startup grant was given to purchase equipment for spin-off commercialization and scale-up production of Nutrio. The product is patented by IPOPIL with certification patent number 1-2017-000199. And Nutrio has a full registration from FPA for annual and biennial industrial crops with FPA product registration 6445. And finally, commercialization of Nutri-Foliar Spray biofertilizer for sugarcane. By demographics, the target core market are the medium-scale sugarcane plantations with areas ranging from 10 to 50 hectares. And the secondary farmer planter clients are the small, ranging from 1 to 9 hectares, and large plantation ranging from 51 to 100 hectares or more. Through the DOSTP card startup program, Fulmite Agricultural Corporation was given the privilege to procure equipment urgently needed for scale-up production of Nutrio for sugarcane. Currently, Fulmite Agricultural Corporation is promoting Nutrio for sugarcane. Feedback from sugarcane farmers are very encouraging, manifested by their repeated orders of Nutrio.
Siyempre, yung NutriBio na malaking tulong kasi yung farm inputs mo, mababa lang, and then ganito kalaki yung kita mo, di ba? Malaking tulong talaga siya, ma'am. Actually, I'm Sander Manalo, engineer by profession actually, electronics communication engineer. I live in Capas, Tarlac. Okay, I'm 37 years old. Actually, father ko is a farmer, pero by profession, civil engineer din siya. No? Yun yung hirap kasi nung buhay dati. Sabi niya kasi nakapag-aral lang siya due to farming, and then nag-invest siya ng mga lupa. So, naiisip ko among 5 of us na siblings, no? Walang magma-manage ng mga naipundar nila. So, yun yung isa sa nag-trigger sa akin para yung farming pasukin ko din. Way back December of 2021, no? Talagang hintuin ko na ba yung sugarcane farming? Sabi ko kasi almost kung hindi kami na break even lang, lugi pa ng thousands. So sabi ko sa misis ko noon, okay pa ba tayo sa sugarcane? And then gladly, merong isang training nga doon sa DOSTP card. Sabi niya, itong NutriBio, pwedeng i-try. So, tinry ko naman yung 4 crops. Nakipag-coordinate ko doon kay Dr. Padilla. And then, I'm so glad naging overwhelmed naman yung support niya sa akin as a new, kumbaga, dito sa gamot na to for NutriBio. So, malaking tulong itong Nutrio kasi it lessen yung expenses, no? Yung farm inputs namin doon sa sugarcane. And this year, nung na-apply ko yung NutriBio, actually, roughly 100,000 yung nasave namin. Fertilizer farm inputs sa doon lang yun, ha? Wala pa yung manpower na para i-gamitin. Unang-una, no? Dati, sabi ko nga, sa isang furrow, kung medyo marami na siyang tiller, okay ka na. Pero ngayon, dahil sa NutriBio, sa sobrang dami ng tiller, hindi mo na mapasukan eh. Okay, so, ang ganda ng feeling, no? As a farmer, as a sugarcane planters, no? Laking tulong talaga ng NutriBio kasi nga napapagaan din yung kalooban namin.
Parang naging worth it yung effort.
Yeah, actually, biruin mo sa dami, ma'am, ng gastos, no? At least nakikita mo na napoproject mo na may mapupuntahan yung ginastos mo dun sa maintenance pa lang. Ayun, ma'am. Actually, siguro roughly mga 300 to 500,000 siguro yung makukuha.
Yung forecast?
Yeah, forecast namin. For planting season lang ngayon na based dun sa kumbaga current price market ng sugar, ma'am. Okay, yun nga, bago kayo malugi, mag-Nutrio na kayo.
Aside from sugarcane, there are some farmers who tested Nutrio on other agricultural crops such as rice, corn, and leafy vegetables. Please watch the testimony of a rice farmer from San Fernando, Pampanga.
Nakita ko yung timbang kasi ang timbang nung without Nutrio nasa 53, 54, 55, ganon. Pero nung nag-spray na kami ng Nutrio, ang timbang niya, nagulat ako, nasa 62, 63, ganyan, may 67, 68. Ako po si Joanna Manansala. I'm 44 years old from the city of San Fernando, Pampanga. So, dahil ang husband ko tsaka ako, we're very interested to acquire a land for our retirement. It's actually a retirement plan. So, nakahanap kami ng lugar dito sa Florida Blanca. And dahil na-inspire kami sa kaibigan niya, so sabi namin, magtanim din kami ng mga gulayan, mga iba-ibang crops, tulad ng sili. So, kaya, that's how we got into farming. Through a friend, inspire kami sa stories nila, and we want to do the same and also be able to help farmers also to support our local farmers. Kaya kami napunta ng farming. Nakita ko yung bulas ng palay. Ang ganda ng dahon, makapal, tapos ang ganda ng pagka-green. Tapos, malalapad yung mga dahon niya. Mas malaki ang kita, definitely. Dahil mas mabigat yung timbang ng palay per kapal versus dun sa mga, nung hindi pa ako gumagamit ng Nutrio, makikita ko talaga yung difference sa timbang. Tapos, maganda yung grains, malinis, malusog. Kaya malaki talaga yung difference nung pag naglagay ka ng Nutrio versus dun sa wala kang supplement ng Nutrio sa palayan at sa kahit anong gulay. Personally, ako I recommend the use of Nutrio sa inyong mga crops sa farm, palay, gulay, prutas. Kasi talagang malaking natutulong niya para mapatibay niya ang mga halaman. Sa kahit anong peste or sakit or panahon na masamang panahon, talagang napapatibay niya yung halaman. So, kailangan niyong lagyan ng Nutrio ang inyong mga crops sa farm bago pa ang sakuna, mag-Nutrio ka na.
Fulmite Agricultural Corporation have already applied for label expansion of Nutrio for rice, corn, and leafy vegetables at Fertilizer and Pesticides Authority to cater those farmers who tested Nutrio on the above crops mentioned. Label expansion of Nutrio for rice, corn, and leafy vegetables at Fertilizer and Pesticides Authority will take 3 more separate bioefficacy trials for the above-mentioned crop for another 5 to 6 months. Based on the experience of Fulmite Agricultural Corporation, the stories we heard from farmers and those who have repeated orders for almost 4 years since they started using Nutrio for sugarcane are very encouraging. I would like to take this opportunity to thank the funding support of DOSDP CARD and the implementing agency, Biotech, University of the Philippines, Los Baños, and the Pulmite Agricultural Corporation as the spin-off company. For more information about Nutrio, please contact Pulmite Agricultural Corporation on the following mobile numbers: 0917-1798-542, 0917-584-1412. We also have email padilla7.jaime@gmail.com, bmpadilla54@gmail.com, virgin.padilla@gmail.com, our website nutriofoliar.com, and our Facebook at nutrio.official. Thank you very much for listening.
Thank you so much for sharing Nutrio Biofertilizer with us, Ma'am G. I'm sure malaki ang maitutulong nito sa ating mga farmers. Again, that was our first speaker, Dr. Virginia M. Padilla, with her talk on high yield at lower cost with Nutrio Biofertilizer for sugarcane. Now, if you have any questions regarding Nutrio Biofertilizer, just leave your questions or comments in the comment section. Remember to state your name and affiliation or location before posting your question. Okay, so now let's move on to our second speaker. Our second speaker is a professor at the College of Veterinary Science and Medicine of Central Luzon State University, or CLSU. She pursued a Doctor of Public Health at the University of the Philippines, Manila. She specializes in the field of parasitology, zoonosis, clinical pathology, foodborne diseases, and molecular diagnostics. She is also the founder and director of the Blitzkrieg Animal Disease Diagnostic Center. In addition, she recently received the DOST SAT Award as one of the startups of the year 2022 for the development of the Andali rapid test kits intended to control swine fever in the country. With her talk on the Nanogold Biosensor Test Kit for African Swine Fever, or ASF detection, let us welcome our second speaker, Dr. Clarissa Yvonne J. Domingo. Ma'am Cla?
Hi. Hello.
Hi, Ma'am Cla. Kamusta naman po kayo, ma'am?
Yeah, I'm okay. Well, I would like to share the video that I would like to share to the listeners. I hope they will watch and learn about the test kit on African swine fever that my team and I developed.
Okay, ma'am. I'm sure our viewers are very to know about this. So, ma'am, how fast can the nanogold biosensor test kit detect the African swine fever or ASF virus?
Okay, it only takes 35 minutes. Yeah,
So, ganun
and
kabilis po pala, madedetect agad. Okay, so I guess I won't keep you for long na po, ma'am. I'm sure our viewers are already thrilled to hear more about the nanogold biosensor test kit. So, take it away, ma'am Cla.
Okay, thanks. So I hope you will watch, listen, and learn. Good afternoon to everyone. I am Dr. Clarissa Ibon Domingo, a professor of the College of Veterinary Science and Medicine of the Central Luzon State University. I will be presenting the results of the development and validation of the Nanogold Biosensor Test Kit used for decentralized ASF viral detection with the aim to empower the animal health practitioners and farm owners to spot the infection among asymptomatic pigs in order to implement wise and timely control and preventive measures within the farm. Last May 11, 2021, President Duterte issued Proclamation number 1143 that places the entire country under a state of calamity due to African swine fever. This warrants an urgency to jumpstart the rehabilitation of the local hog industry towards the availability, adequacy, and affordability of pork products for the purpose of attaining food security. The Department of Agriculture launched the Bantay ASF sa Barangay, popularly known as Babay ASF, and a twin program known as INSPIRE, which means Integrated National Swine Production Initiatives for Recovery and Expansion, with the goal of instituting biosecurity and disease control measures in tandem with calibrated repopulation and enhancement of the local swine industry. In confronting the challenges, CLSU developed the ASF Nanogold Biosensor Test Kit, which combines the loop mediated isothermal amplification and hybridization of a probe and gold nanoparticles to detect the viral genetic material taken from the animal or from the environment. This is the framework of how the test kit was developed. LAMP primers were designed based on the nucleotide sequence of the index cases in Rizal and Bulacan in 2019. Optimization for the LAMP protocol using the DNA extracted from the specimens by the modified solding out method and hybridizing the amplified LAMP products with a probe coupled to gold nanoparticles will cause aggregation with the amplified products to show a change in color. After analytical sensitivity and specificity were done, the laboratory workflow was assembled into a practical, simple, and easy to use test kit that required simple, unsophisticated laboratory equipment such as a heat block and a microcentrifuge. This slide shows the phylogenetic analysis of the detected Philippine ASF isolates from different specimens such as organs, like the spleen, blood samples, surface swabs, water and fecal samples. All belong to the most prevalent genotype in Eurasia, which is the genotype 2. By the way, there are 24 ASF genotypes reported throughout the world. In the Philippines, there are two strains, genotype 2A isolated in 2020 and 2B from 2019 specimens. This slide shows how the test kit is used. DNA is extracted from the specimen using the modified soling out method. The DNA extraction takes 1 hour and 30 minutes from the receipt of the specimen up to rehydrating the DNA pellet ready for amplification. Then, 1 drop of the rehydrated DNA pellet is instilled into a 1.5 ml tube containing the LAMP nanopremix. This tube contains the BST enzymes, primers, and the buffers for amplification. The tube is inserted into the slot of the heat block for amplification at 58 degrees centigrade for 30 minutes. At the end of 30 minutes, the tube is removed from the heat block. A foil which contains the G-dye tubes is unwrapped, and one of the G-dye tube is opened and all the contents is aspirated and added into one tube that was removed from the heat block. The G-dye contains a special probe and the synthesized gold nanoparticles. After all the tubes receive the G-dye, these are inserted back to the heat block for another 5 minutes at 58 degrees centigrade. This will stimulate hybridization of the probe and the gold nanoparticles to the amplified LAMP products. After 5 minutes, a color change will be immediately observed, and the reading is by colorimetry. One kit box contains canisters for DNA extraction, such as the lysis buffer, extraction buffer, wash buffers 1 and 2, and the rehydration buffer. Consumables are also included, such as microcentrifuge tubes, mini droppers, tuberculin syringes with needles that replace the micropipeters, and rubber floaters. The box also holds PCR tubes that contain the G dye. These tubes are wrapped in aluminum foil. The G dye tubes can be stored at room temperature away from light or inside the refrigerator at 4 degrees centigrade. There is a separate container that holds frozen gel coolants, or what we call cold dogs, that help preserve the tubes that contain the lamp nanopremixes. These lamp nanopremixes must be stored in the freezer at negative 15 degrees centigrade to lengthen their shelf life and to prevent self-annealing of the primers due to the presence of the enzyme BSD polymerase. Only when testing is ready shall these lamp nanopremixes be removed from the freezer for use. Analytical specificity was done to assess if the originally designed primers, based on the Philippine isolate, could differentiate the target gene, which is P72, from non-target genes of other viral isolates, such as the classical swine fever or hog cholera, PRRS, swine influenza virus, and rotavirus. The positive reading is seen as wine red color, while the negative reading is seen as purple to gray. Analytical sensitivity was done to assess the lowest serial dilution of the DNA concentration of the positive specimen if it can still produce multiple ladder-like bands in the gel after electrophoresis, or a 250 base pair single band after PCR. In the nanogold biosensor, the lowest detection limit of the serial dilution is at 10 to the negative 4, while in the PCR it is at 10 to the negative 3. Here it is already observed that the lamp amplification of the nanogold biosensor is more sensitive than PCR. Likewise, the intensity of red at 10 to the negative 4 dilution is the last before the color turns into purple. This table shows the persistence of the virus remaining stable and infective in different specimens and temperature. One can see that the virus survives longer in organic matter on surfaces. Hence, the nanogold biosensor can detect the viral genetic material on environmental surface swabs, water, feces, pork meat, whether chilled, fresh, or even processed, and blood or serum. This table shows the 9 feed ingredients where the ASF virus can be recovered. Hence, the nanogold biosensor can be used on raw materials or commercially mixed feed preparations to detect the viral genetic material. These are the two package formats of the test kit. The left one is the box being manufactured and commercialized by a tech startup company founded by the developer, while the right box is the packaging distributed to the DA regional field offices during the Babay ASF and Inspire programs, in which the production cost was funded by the Department of Agriculture. The pictures are the different specimens or samples that can be tested using the test kit. Specimens can be taken from the animal or from the environment. Blood specimens can be collected by piercing the ear vein of the pig and allowing the blood to be absorbed by a sterile cotton wad. There is no need to collect from the jugular vein of the pig to reduce invasiveness. The blood-soaked cotton wad is placed in a clean Ziploc plastic bag, labeled properly by a marker pen, and transported in a cold storage box to the laboratory. For fresh or chilled raw pork, homogenize by cutting finely and squeezing with sterile scissors and spatula to extract the meat juice for DNA extraction. For processed meat, wash to remove all the spices and sauce. Homogenize the meat to extract the juice for DNA extraction. This table shows the competitive advantage of the nanogold biosensor test. Amplification reaction takes 35 minutes as compared to conventional PCR and ELISA. Equipment required to use the test assay efficiently is lesser and more simple as required by PCR and ELISA. The BST polymerase enzyme in lamp amplification is not affected by contaminants as compared to ELISA and PCR, which are prone to inhibitors, hence are liable to give false results. The cost of one test is more affordable than PCR and ELISA. Another important advantage is that the test has its own DNA extraction steps, whereas PCR requires an external commercial extraction kit that is expensive and is limited to a number of samples only. During the launching of the Babay ASF, the Nanogold Biosensor test kit was used to assess the biosecurity readiness of farms to accept reloading of sentinel animals. The test kit was also adopted by the Provincial Veterinary Office of Iligan City and Cabacan, where the Veterinary Hospital of the University of Southern Mindanao was responsible to test surface swabs collected and submitted by the MAO and PBO. These are the different provinces where the staff of the Provincial Veterinary Office conducted surface swab collection among smallhold hog farmers in the Babay ASF and Inspire programs of the Department of Agriculture. This slide shows the surface swab results in both biosensor test kit and PCR from a commercial and smallhold farms heavily hit by ASF. All the pigs were stumped out and the farms were left vacant for 1 to 3 months. Biosecurity profile was done using the biosensor and conventional PCR prior to loading with sentinel animals. Results showed that the biosensor test kit detected more positive reactors than the conventional PCR using the surface swabs. Hence, a thorough, careful disinfection was immediately conducted in these farms. As you can see, the positive reaction in surface swabs is color gray, while the negative reaction is color red, which is entirely opposite for the samples in blood and tissues. The water samples were tested also because ASF virus has been reported to survive 2 months in water. The samples were collected from the main line before distributing to the different houses in the farms. Again, biosensor detected more positive reactors than PCR. Positive water samples came from farms with history of burying dead pigs within the farm. In this table, fresh and old nanogold fecal samples were positive in both the biosensor and PCR tests. But for the saliva samples, biosensor can detect the virus better than PCR, even if the same pig was positive in both tests for its blood and feces. From the time the nanogold biosensor was included in the Babay ASF program in February 2021, slowly the red zones in the ASF zoning map changed colors, and the status by September 2021, as reported by Dr. Ruth Sonaco, the director of the National Livestock Program, is shown in the picture. Cities or municipalities with no reported ASF cases beyond 90 days changed from red to blue, and beyond 180 days to gray. Impact showed an increase from 9.7 million to 9,866,446 heads in October 2021. The benefits of the nanogold biosensor test can be deployed to other Asian countries suffering from ASF. Validation for product registration conducted by the Bureau of Animal Industry ADDRL using surface swabs showed that the sensitivity of the nanogold was 100%, but the specificity was only 41%. The reason for the low specificity could be due to no established cycle threshold values of the quantitative or real-time PCR for surface swabs, since the real-time PCR protocol was originally made for blood samples only. Likewise, to ensure the fairness of evaluation, Fu et al. In 2020 said that both the primer nucleotide sequences used by two different assays must be designed to bind on similar location of the target gene. In this case, the ASF nanogold biosensor lamp primers were designed based on the Philippine isolate in 2019, whereas the BAI qPCR primers were designed based on the Georgia strain in Eastern Europe in 2003. The Bureau of Animal Industry ADDRL validation using the blood showed even a lower sensitivity and specificity of the nanogold biosensor because the ADDRL's real-time PCR used RNA blood extracts while the nanogold used DNA extracts of the blood. Likewise, the primers were designed based on different location of the target gene. However, when all the blood samples were extracted for DNA using the same modified sorting out method of the nanogold biosensor, the sensitivity and specificity values improved. Nevertheless, since the primers were still different, the accuracy percentage of the nanogold pitted against the real-time PCR assay of the ADDRL was only 75%. When the nanogold primers and the DNA extracts were assessed using real-time PCR conducted at CLSU, sensitivity and specificity values of the nanogold biosensor on all types of specimens were above 85%, reaching up to 100%, with accuracy percentage of above 87%. Using the DNA extracts, the F3B3 lamp primers of the nanogold biosensor test performed better in conventional PCR than the BAI primers. On the other hand, the BAI PCR primers performed better on RNA extracts than the F3B3 lamp primers. Meanwhile, the lamp primers in the nanogold biosensor test show consistent performance in amplifying the target gene on either RNA or DNA extracts, thus outperforming the BAI PCR primers. One important reason is that there are three pairs of lamp primers in the nanogold biosensor amplification, which makes it more sensitive in amplifying the target gene, whether in the RNA or DNA extract forms. Table 7 shows the versatility of the nanogold test kit because it can amplify the viral genetic material from different types of samples. Results also imply that pigs sent for slaughter are positive for African swine fever. Another observation was that the positivity rate is higher in the nanogold test kit compared to the conventional PCR. This implies that the ASF test kit is more sensitive than PCR. It is now implied that pre and post slaughter examinations for African swine fever viral detection must be a routine practice in the abattoirs using the nanogold biosensor test kit to mitigate the spread of the infection along the food chain until the infected pork reaches the chance to be rendered as swill feeds or raw protein materials in commercial feeds. Likewise, the positive surface swabs collected from hauling vehicles of live pigs could be potential spreaders of the infection. Also, the nanogold test kit was able to amplify viral genetic materials in commercial feeds. Further, there were more commercial feeds contaminated by the African swine viral genetic materials tested in the early 2021 as compared to those in early 2022. Unfortunately, there was no attempt to identify which raw material in the feeds was contaminated with the virus. Finally, the nanogold test kit was able to amplify the viral genetic material in water samples taken from 82 farms. These water samples were taken from the deep well of the farms in early 2021. Results showed that the ASF positivity rate in water using the nanogold biosensor test kit was 81%, while conventional PCR 23% only. This implies that the groundwater could have been contaminated when dead pigs were buried in the farm premises without the proper ground preparation of putting a lining to prevent leachates from reaching the water table. As the carcass decays, the virus reaches the water table through seepage or leaching. As a conclusion, the ASF nanogold biosensor test can help assess freedom from 1. Contamination in your farm pens, water source, PPEs, farm tools, and farm vehicles. Freedom from worry about infection among your animal stock. Freedom from feeding contaminated feeds, and freedom from consuming contaminated raw and processed meat. By decentralizing diagnosis using the ASF Nanogold Biosensor Test Kit, every animal health practitioner and farm owner is empowered to do early detection towards a wise and timely implementation of control and preventive measures within their farms. The test kit is good for 10 sample reactions, and one reaction costs only 400 pesos, or an equivalent of 6.80 US dollars. To sustain the production of the test kits, the project has tapped an earth enterprise that pioneers in the commercialization of Philippine-made rapid test kits for transboundary animal diseases. The developer of the kit would like to acknowledge God Almighty for providing her with wisdom and guidance throughout the development of the test kit, and DOSTP card, whose continuous support to budding and seasoned researchers is beyond compare. Thank you.
Thank you so much for your presentation, Ma'am Cla. Again, that was Dr. Clarissa Yvonne J. Domingo with her talk on the Nanogold Biosensor Test Kit for African Swine Fever or ASF detection. Now, with two presentations done, we have one more to go. But before that, again, let me remind our viewers to post your questions in the comment section, especially those regarding Nanogold Biosensor Test Kit in our Facebook Live. And moving on to our third speaker. He is a retired researcher and now a private consultant from the University of the Philippines, Visayas or UPV. He holds a PhD in food process engineering and specializes in post-harvest fisheries and marine bioactive compounds. He was awarded by UPV the 2021 Chancellor's Award on the Most Outstanding Achievement in Research and recently received numerous international publication awards by DOST Picard. To discuss the utilization of marine fisheries species in the production of protein hydrolysates, we are all very pleased to have Dr. Jose P. Peralta. Doc JP? I think Doc JP, kindly unmute, sir.
Yes.
Yan, welcome, sir.
Good
Kamusta
afternoon
po,
to
Doc? Kamusta po
yes,
kayo?
pleasant good afternoon to you, Miss Danica, and a pleasant good afternoon to all our viewers who are participating in this biotech conference.
Thank you, sir. Sir, I think do we just have a question for you, sir. I think our
Go ahead.
viewers would also be curious about this. Are the products that you're going to share with us today safe to consume?
Oh, they are. In our process protocols, in the way we manufacture, we already replace those chemicals that are not food grade. And the chemicals that we use are all food grade. And at the same time, we conducted a toxicity test and they all passed the toxicity test, as will be mentioned later in the presentation.
Okay, thank you, sir, for enlightening us. I think our viewers are excited na po para mapanood yung presentation niyo. So, take it away, sir.
Thank you. My the product that we are developing are basically proteins or pre-digested proteins. This is similar to milk products that is in powdered form. So hindi ka nakakain, simply get two spoonfuls of this protein hydrolysate powder, mix it with water, hot or cold, and then simply drink it. So parang kumain ka na. And with that, as an introduction, here my presentation on marine fish hydrolysate products and project. Take it away, Dan. Good afternoon, ladies and gentlemen. My presentation is about protein hydrolysis from marine fisheries species. According to Hippocrates, Greek philosopher, let thy food be thy medicine and thy medicine be thy food. So even at that time, food is the key for our wellness. The functions of food. In the old concept, the functions of food are for energy keeping, bodybuilding, and protective mechanisms. These are supplied by what we eat in terms of fat, carbohydrates, protein, minerals, and vitamins. In the current concept, we have the primary function, which is the nutrition function, to nourish the human body, and the secondary function, the sensory function, to provide palatability and emotional satisfaction, and the tertiary function, the regulatory function, to act as regulator to the physiological systems of the human body, regulation of basic metabolic processes, the regulation of oxidative stresses, regulation of gastrointestinal physiology, and the regulation and modulation of the immune functions. associated to a variety of names: health foods, nutraceuticals, food supplements, wellness foods, fabricated foods, or even designer foods. Different terms leading to one message, that is, foods that are efficacious in the promotion of optimal health for the prevention and treatment of diseases. If we look at this figure, the mortality, 10 leading causes of mortality in the Philippines in 2019, i- ischemic heart diseases, cerebrovascular diseases, diabetes mellitus, hypertensive diseases, and other heart diseases. Uh, Most of these can be prevented by proper diet and proper lifestyle. The same is true in 2021, last year, and currently, January, February of 2022. The leading cause of mortality hasn't changed much. The project direction that we are pursuing is to develop nutraceutical products to enhance the regulatory functions of our body metabolism, to develop pharma-grade products that enhances the immune system for its modulation enhancement of body metabolism, to establish a startup company to pursue these research directions. As a background there, protein hydrolysases are used for the nutritional management of individuals who cannot digest intact proteins. These are post-surgery patients, persons with health dysfunctions like uh, people with cancer, uh, patients with hypertension, diabetes, and the like. Uh, Hydrolysates provide sufficient nutrition for health maintenance of health-impaired individuals. The hydrolysate can also enhance and alter functional, physiological, and sensorial attributes of the proteins without compromising its nutritive value. First off, by definition, protein hydrolysases are pre-digested proteins. Other terminologies used in the presentation as well as in the project, when we talk about surimi, these are comminuted fish proteins, normally as a result of flesh bone separator machining, or simply put, these are blended proteins. Hydrolysates from fish, these are fish proteins that has been broken down into smaller molecules or simply pre-digested proteins. So from the protein, it becomes peptides, it becomes amino acid, and when the amino acid degrades, it becomes ammonia. The project objectives in general is to develop products from marine resources that are easily digestible with full nutrition and would complement sickness healing. Specific objectives, these are to refine the process protocols and increase raw material sources from marine resources, including that of fish and mollusk species. Blend different fish hydrolysates from different fish species for an appropriate product mix. Conduct and determine bioactivities of the blended products. Protect the knowledge generated via patents and copyrights. Further, blend fruit berries, extracts, and concentrate just to mask the fishy odor and taste, and conduct acceptability sensory tests on the formulated product blends. Produce nutraceutical from hydrolysates and conduct toxicity tests, safety tests, as well as market study of the products developed. And finally, to initiate sustainability issues by using cultured species as raw materials. In year one of accomplishment, we focus on the optimization of methods, production of protein hydrolysates, and functional characterization. The functional properties sought for were solubility, bulk density, absorption capacity, all absorption capacity, hygroscopy, water activity, and moisture content. The marine species used were oyster, Crassostrea iridale, big ice cod, sardines, mussel, bullet tuna, sea bream, snake mackerel, island mackerel, purple back squid, and that of seaweeds. The established assay and production protocol used. These are the standard laboratory protocols from published literature indicated as modified should there be modifications made. As listed, this is on water absorption capacity, antioxidative activity as a result of DPPH radical scavenging activity. And in this slide, we have the conversion processes. Starting from raw material, it's clean, and most of the muscle protein are separated from the skin, bones, scales of the fish raw material. Pre-treatments are made, and we isolated the protein via pH chip method, as well as comminuting the uh, fish flesh. Thereafter, we proceed to enzyme hydrolysis using specific enzymes like pepsin, bacillus enzyme, as well as bromelain, resulting to liquid hydrolysate. And this liquid hydrolysate are further dried either by spray drying or freeze drying. In the year two accomplishments, we focus on substituting chemical grade ingredients to food grade ingredients to produce a food grade protein isolates and hydrolysates. So at the end of year two, we already satisfied the objective of producing hydrolysates from the 10 raw material as mentioned. Some of the pictures will be shown. These are the hydrolysates from sardines and oysters. If you will take notice on the hydrolysates produced, these that of sardines are a little bit lumpy. We found out that our hydrolysates produced are hygroscopic. It easily attracts moisture from the atmosphere. These are hydrolysates produced from big-eyed scud as well as purple back squid. They develop products accordingly after defining the hydrolysates from the raw materials as discussed. We blended and came up with a core hydrolysate blend. From the 10 hydrolysates, certain proportions as provided by the amino acid analysis, we mix them together to produce a core hydrolysate blend mixture. And then from the core hydrolysate blend mixture, we added some ingredients for stability, product stability of the powder, as well as palatability, addressing sensorial attributes. So we came up with a final product blend formula. In year three, our work focused on the amino acid profiling as well as formulation, sensory analysis, and functional characterization of the product. We also conducted food safety assays. These are pH, aerobic plaque count, moles and yeast count, water activity, as well as toxicity measurement. Functional characterization made were moisture content, soluble protein, water activity, hygroscopy, bulk density, water absorption capacity, as well as oil absorption capacity. In addition to that, we did conducted a marketing report that includes market analysis, market study, and forecast. All of these are not as exciting if we did not conduct biological activities like antioxidation from DPPS RSA, ABTS RSA, hydrogen peroxide scavenging activity, and antibacterial activity, as well as anti-hypertension activities. The results of our say suggested the past food safety and toxicity test. We have characterized the physicochemical products attributes of our products. The bioactivities we conducted, it's present, all right, and we even measured the bioactivities. These are the antioxidants provided for by the DPPH, RSA, ABTS, RSA, and H2O2 scavenging activities. As antimicrobials, we detected presence of antimicrobials, and we even detected antihypertension. This is the ACE or angiostensin converting enzymes inhibition activity. Further, we compared the hydrolysate produced relative to our standard, which are egg albumin and dried egg white. It has a total energy value in kilocalories of 344.93 and 382 kilocalories. The hydrolysates from black scabard fish, lowfin pomfret, bullet tuna, mussel, as well as oysters do have total energy value really comparable to that of the standards. These are some of the images in the laboratory working on our research activities. And as far as IPR, we have registered 2 IPR. That's part, the title of which are fertilizer from seafood and animal feedstock supplement from seafood. Third one is in the process. It's entitled Fish Hydrolysate Process and Fish Hydrolysate Product. We have accomplished 3 publications in Elsevier Web of Science recognized journals. The first one is entitled Production and Characterization of Protein Isolates and Hydrolysates from Sleeper Cup Oyster. The second one is entitled Changes in the Physicochemical Properties and Antioxidant Activities during reduced salt fermentation of green mussel. And the third one is more on looking at the value of the fish hydrolysates as feeds. And somehow we have encouraging results. We also made posters defining our utility model IPRs during one of the exhibitions of the College of Fisheries and Ocean Sciences. In terms of products that we have established as prototypes, we have 10 protein hydrolysate powder from the 10 fisheries species, one core blend product powder, which is a mixture of the 10 hydrolysate of different proportion according to their amino acid profiles. And finally, one blended product powder, core blend product powder added with other ingredients to somehow enhance palatability as well as sensorial stability. We encourage participation of Master of Science students in our project, wherein we have 2. We encourage participation of faculty members. Actually, they were not faculty members before, but upon graduation from the MSP series program, they were hired accordingly. Lumen Tejano, now faculty assistant professor of UP Visayas. Anilin Gomez, faculty of Cebu Technological University. And Franz Vergara, a faculty, now faculty of Partido State University. We also train researchers, 3 of them, as well as supported the training of personnel in project administration. In the course of the 3-year activities of the project participated in the first Cradle Symposium in last January, as well as obliged project interview with TSME Homebody kasama si Jamie Quinto last January 13, 2022. The video files of these events can be seen in the YouTube archives. Further, 3 students from Taiwan spent their summer internship at the College of Fisheries and Ocean Sciences, specifically at the Institute of Fish Processing Technology. Our project participated in these events. This is what we have come up thus far, our project video.
Hi, I'm Dr. Jose Caranta, the initial project leader of the Cradle Hydrolysis project. The project came about as a long-term activities. The intent actually is for the industry to have a confidence in the institution, the Institute of Fish Processing Technology, to do a commercial research for them. Department of Science and Technology, the research and development arm of the Philippine government, took note of the initiative. Whatever we have accomplished in the USAID project were continued in the Cradle Hydrolysis program. The agenda is to bring the industry closer to the academe, such that the industry will have its confidence in the academe, and the academe will have its expertise and capability to provide what the industry would want.
How do we raise the level of science and find new ways to determine the best model of discovering natural product-based medicines? In order for us to do things better, we're collaborating with public institutions or state universities and colleges like the University of the Philippines and its different campuses. We're collaborating with private institutions like Ateneo de Manila, the Technical Institute of the Philippines, and we're getting grants from the Department of Science and Technology, but also the Commission on Higher Education through its PICARI, through its Philippine California Advanced Research Institutes program. The platform that we've established through Core Lab, it's an open innovation platform, meaning come with us with your innovative ideas. Let's engage in research together. That's how we conceived the Partner With Us campaign.
I am Lumina, the current project leader of the DOSCP CAR project entitled Protein Hydrolysis from Marine Fisheries Species under the Cradle program. The project aims to produce a product that addresses current issue and provide a better therapy food because of the production of bioassimilable and bioavailable proteins and bioactive peptides from marine organisms. This can be achieved through the production of protein hydrolysis, which can be a good source of bioactive peptides and proteins. We will also determine the biological activities of this product mix. Our experiments mainly focus on the production of protein isolates and protein hydrolysis using food-grade reagents.
What is the importance of this initiative in the growth and development as an institution of UPT Science? We have started this collaboration with the industry. All of these initiatives, the Institute of Fish Processing Technology is taking the lead. The collaboration with Pascual Pharma Corporation under the Cradle program to crystallize that need to have a long-term relationship with a industry partner engaged in a research collaboration of their interests, provide a product that will be able to be commercialized by the industry. On this DOSC Cradle program, we are now tracking on year 3. Our progress on this marine peptide hydrolysis is in accordance to the time frame that we have promised the UST. After year 3, we will be able to produce products that is worth pursuing we will be able to protect this under IPR arrangements, and we will be able to tell potential investors that this is the potential of the said products. The recommendations and next steps, given 3 more years, the project would have commercialized nutraceutical products. Given 15 or 17 more years, inclusive of clinical trials, phase 1, phase 2, and phase 3, the project will be able to come up with pharma-grade products. For anti-COVID-19 virus preventive therapy, the products that we are endeavoring has a lot of potential. And a startup company will be established to manage the research and commercialization thereto. So investors are welcome to negotiate. That's all for today. Marami pong salamat sa inyong pakikinig.
Thank you, Doc JP. Again, that was the utilization of marine fishery species in the production of protein hydrolysates by Dr. Jose P. Peralta. And there we have it. Once again, those are our 3 presentations on the biotechnology applications in the agriculture and aquatic sectors. And now I think we already have our questions in the comment section. And with that, let me call on the screen again our speakers, Ma'am G, Ma'am Cla, and Sir JP for our open forum. Hello. Hello po, Ma'am G, Ma'am Cla, and Sir JP.
Yes.
Yan.
Medyo natabunan ako.
Let's see. Alright,
Okay.
ayan, kitang-kita na po yung mga faces natin. So, ma'ams and sirs, before we begin, I would like to ask, handa na po ba kayo na sagutin ang questions ng ating mga viewers? Ma'am Cla, handa na po ba kayo?
Yes.
Yes.
Handang-handa na.
Sir JP?
Take it away, Miss
Okay.
Dan.
Okay, so we like that energy po. So, for our first question, this is from Miss Lynn T. Luzaran. So this is for Dr. Padilla. Ma'am G, good afternoon. I am Lynn Luzaran from the Philippines Sugar Research based in Negros. So her question is, herbicide is being massively practiced in Negros in mostly all farms due to the scarcity of farm labor. It is used as a pre-emergent and as a post-emergent, and usually 4 times during the interduration of sugarcane crop. So with this practice, will it affect the Nutrio application?
Thank you for that informative question. First of all, Nutrio is applied only 2 times. That is 2 months after planting and 4 months after planting. So if that there would be no problem with pre-emergence and also for post-emergence because at 2 months the plants is well already established, and 4 months only twice ang application ng foliar by spraying. So if it is applied by spraying, kung mapapatiyempo siya doon sa time ng pag-spray with Nutrio, ang ano lang doon ay it should not be mixed together with any herbicide. And besides, Nutrio should be sprayed only using solely for Nutrio, not those sprayer that have been used for herbicide and insecticide. Cannot be mixed with insecticide or herbicide because the microorganism in Nutrio are live microorganisms. That's why it is applied alone.
Okay, thank you po, Ma'am G, for answering the question from Ma'am Lynn. So let's move on to our second question. So our second question is from Ms. Diane Villamora. So this is for Dr. Domingo. So what are the criteria that should be considered to ensure fairness in evaluating a new test assay?
So, first of all, when the approach in testing or doing parallel testing with between a new test assay and with that of the current existing assay na supposedly this will be used as our referent or reference test, ano. So, it should be made in an integrated approach. First, uh, if we're going to use qPCR or real-time PCR, the CT values should be standardized based on the sample that is going to use. So we cannot use a cutoff value or a CT values of qPCR when it is originally made for blood. And what you're going to do is to test a new assay that is intentionally used for another kind of specimen. So, dapat parehas sila para matanggal ang bias. Second, if the molecule to be used is DNA, DNA lang for both. Or RNA lang for both. Hindi pwede yung isa DNA, yung isa RNA. Kasi there's another bias for that. Third, the primers that should be designed on the same locus or location of the target gene. So, if, for instance, the same nga ang target gene, let's say sa ASF is P72, but the other one has a different location as that of the other one, let's say, no? So, hindi rin pantay yun. Dapat parehas. So, this is an integrated approach. The fourth one is that, the primers, no? If we're going to test a protocol using, let's say, 3 primers, such as for my test kit, for instance, it uses 3 pairs of primers because it works on the principle of loop-mediated isothermal amplification or LAMP. And whereas for the real time, it only uses 1 primer, which is 1 pair of primer, no? The forward and the reverse. Doon pa lang, hindi na rin may bias na rin yun. Okay? So, there are many things to consider. And somebody that I interviewed from Philippine Genome, when COVID was being tested using real-time PCR, they have to train many laboratories to do real-time PCR for COVID. So number one criteria, the machine, the real-time machine should be the same brand. So doon pa lang kung magkaiba na, may bias na naman. So para sa integrated approach, we should minimize the probable or the possible biases so that this will ensure fairness in evaluation.
All right, thank you for that answer, Ma'am Kla. So let's move on to our third question. Oh, so this is addressed to Dr. Padilla again, to Ma'am G. So this is from Director Amir Salamat of the Asia Pacific Cultural Heritage Development, Inc., central office based at Morong, Rizal. So the question is, can Nutrio-Bio fertilizer be used for HVCs, particularly those from upland farming, Why? What are the possible outcomes of our post-harvest of these HBCs? Ayun po.
First of all, we I did not conducted any experiment on high value crops, but because this was developed particularly mainly for sugarcane and eggplant. But there are already farmers who tried anecdotal. They tried to use Nutrio in their high value crops like vegetables, particularly like string beans, in sile, what else? Cucumber, even banana and papaya. Those high value crops had been tried by farmers, and it was found na only anecdotal. It is working well with those crops. So for upland crops, I think it will work. Even for rice, as I have shown in the testimony of a farmer who tried to use it for rice.
Okay. Okay, thank you, Ma'am G. And then now for our fourth question. So from Mr. Dexter Buenaventura, this is addressed to Dr. Peralta. So what are the derived benefits of the food supplement if they are consumed? Doc JP? Unmute.
Thank you for the question, Mr. Dexter. Self-serving yung research ko kasi I am already in my senior years and I still would want to live a little bit longer. So I'm trying to make products specifically for senior citizen that is easily digestible. Okay, to the question, derived benefits as mentioned earlier, the fish hydrolysates are already pre-digested proteins. As such, they are easily digested. Inumin mo nga lang, hindi mo nangunguyain, pwede na. Similar to milk, no? Further, in our study, we found out that they contain verified as well as quantified antioxidants, antimicrobials, and even anti-hypertension. There are more actually, pero itong tatlo pa lang yung medyo pinag-aralan namin. That is in accordance to published literature. Furthermore, as a food ingredient for manufactured foods, they show promise as a ingredient stability. Dan?
Okay. Thank you po, Doc JP. Now, let's have our fifth question. So this is from Mr. Paul Fernandez. So this is for Dr. Padilla. Ma'am G, ang question niya po is, does Nutrio biofertilizer have limited application, or can it be applied to all types of plants, trees, or flowers? And magkano daw po?
As I have said before, this is product was developed for sugarcane in the developmental stage. But there are farmers who try this in their crops, even for ornamentals. And I have an experience with in a Sampaguita farmer that have a problem with their Sampaguita because the plants were already deteriorating. So I told her that you cultivate the soil and try to the old leaves so that you can make the plants make it younger, and then spray it with Nutrio. They try it, and they found that it produces very good flowers and more flowers. They prolong the flowering of Sampaguita because according to them, the surrounding Sampaguita farmers, they don't have any more flowers, but their Sampaguita are still producing flowers. And also, there is also a farmer who tried it in sili. According to him, these are all anecdotals. The farmer told me that it promotes flowering and sili prolong the flowering and the fruiting stage for more than a year. So the other farms around the place of his farm have already terminated their sili, but his plants sili, when he continuously sprayed it with Nutrio, they produces more fruits, and he earned a lot from these plants. He covers the lowest price and the highest price of his sili because it prolonged the productivity of his sili. For other crops, flowering, this again anecdotal because I did not have so much time to do the research because I am already retired, and the development of the technology was the time when I was almost retiring. So it's up for the farmers to try it. And as I have said in my presentation, I have applied for expanded label of this Nutrio for other crops like corn, rice, and leafy vegetables. There are already farmers who tried it in leafy vegetables and they found it good, produces. And for string beans, they also produces good harvest. And also for eggplant, even cassava. Well, I don't want to I don't want to start them to try this because I want more farmers to use our product. But of course, we have to follow our government regulations. That's why I have to register and expand the label of Nutrio, because Nutrio can be used for sugarcane, rami, for annual and biennial industrial crops that can be used for this one, even for tobacco. Because in our experiences, those who have tried to spray their plants, even papaya and banana, they produce better harvest when they sprayed it with Nutrio. Again, that is anecdotal. I hope I have answered the question.
Yes. Thank you so much, Ma'am G. I think very clear naman po yung naging answer niyo to the question. So, let's have our next question. So, this is from Dane Perales from Batangas City. So, this is for Ma'am Cla. Is the ASFV nano biosensor a combination of two reactions? Ma'am Cla?
Okay, so the usual, when we say nanotechnology, it's usually a chemical reaction using a probe and metal nanomaterial such as gold or silver or whatever, no? Kung anumang metallic na material for nano. But for the ASF nanogold biosensor, it is a combination of an amplification protocol which uses the loop-mediated isothermal amplification. So parang nucleic acid-based na test wherein, for instance, if the viral load is very low in a particular specimen, it will amplify to make it obvious. And after amplifying, the products of amplification will be hybridized with nanotechnology, which utilizes a probe and gold nanoparticles. That's why nanotechnology enhances the visibility of the amplified products after amplification. So it is now more, let's say, sensitive than the current test. That's it.
All right. Thank you, Ma'am Cla, for answering that question from Dane Perales. Next, I think this will be our last question. So, Our last question is from Mr. Christian Bioneta from Batangas as well. So we are still under the pandemic of COVID-19. So is there a potential use of these products for preventive viral infection? So I think this is for Doc JP.
Thank you very much for that question. There is definitely a potential. The problem is, yung result ng research, kailangan mo 2 years ago. So, we do not have the opportunity to do the research. But be that as it may, our food products, the fish protein hydrolysase, are addressing the third functions of food. They contain bioactive compounds that addresses the modulation and regulation of the immune system. We are addressing the enhancement of technically the immune system. We already know what they are. These are the small molecule functional peptides, about 5 kilodaltons or less. So medyo meron ng kapa kahit papano. There are a lot of literature published citing their value. The project will have to refocus kasi before the pandemic, wala naman antiviral sa agenda ng deliverables namin. Biglang lumabas si COVID-19. Okay, pwede. Kung sa pwede, the point is give us opportunity to do the research. We will deliver according to the time frame that is necessary. Yan. And then we now have protocols in the manufacture of the protein hydrolysase. We can even produce a variety of small molecule peptides needed once we have identified them. We still do not have the protocols to concentrate any specific peptide. We are not into the medical discipline. We are simply food scientists. So we still have to collaborate with the medical team researchers to pursue the agenda. Further, the project need to pursue the phases of clinical trials, phase 1, phase 2, phase 3, for safety and efficacy before they can be verified as effective in preventing COVID-19. Like I've said, there is always a possibility. Which peptides? That is the very big question. We'll have to do the appropriate research.
Okay. Thank you for that, Doc JP. Thank you for answering, ma'ams and sirs, sa questions ng ating mga audiences. And of course, thank you po sa ating mga audiences na nag-send ng kanilang mga questions. And we hope that you were able to get valuable learnings from our 3 speakers who presented interesting technologies that would surely help our farmers. And of course, Ma'am G, Ma'am Kla, Doc JP, thank you for dedicating your time to share with us yung mga technologies na ito. Before you go, we'd like to ask you some few words for our audiences. Ma'am G, do you have any words for our live audiences right now?
Biofertilizer, the sugarcane planter's best friend, for reduced cost of production, increased yield, and income. Sabi nga ay quality fertilizer for quality crops. So for those who want to purchase our product Nutrio, please contact us, Fulmight Agricultural Corporations, as I have shown during my video presentation, the telephone numbers. Thank you.
Okay, thank you, Ma'am G, for that. Ma'am Kla, any words po?
Centralizing the ability to detect or to spot the ASF virus early using the Blitz test kits, every animal health practitioner and farm owner will be empowered to do early detection towards a wise and timely implementation of control and preventive measures to mitigate the spread of ASF. So the test kits, the ASF test kit is an alternative choice as a point of outbreak test kit. Thank you.
Thank you, Ma'am Kla and Doc JP. Doc JP, do you have any words po?
The best we could do for now is really to come up with nutraceuticals. Baka pwede nang i-cover ng nutraceuticals yung COVID-19 preventive therapy. I said baka, so I'm not so sure. Give us 3 more years, we might be able to come up with the nutraceutical products. Medyo maghintay-hintay lang po ng konti. Thank you.
Thank you, Ma'am Ma'am G, and Doc JP. We really appreciate you being here po. So, bye-bye po. Thank you po. Bye po. All right. So, should you have any questions or concerns that you want to further discuss with our speakers, you may reach them through the following emails that are flashed on your screen. And as we are already approaching the end of our program, we would like to refresh your memory on the 3 informative topics on biotechnology applications discussed in this afternoon's webinar. So to give us a brief summary of the 3 presentations, let us call on Ms. Alisa Carol M. Ibarra of the Crops Research Division.
Thank you so much, Dan. Alright, so let me present the synthesis for today's webinar. Good afternoon, everyone. Indeed, what was presented to us today were interesting topics on the biotechnology applications of dust Picard's S&T interventions in the agriculture and aquatic sector. So I would like to thank all the participants who are watching us live via our dust Picard social media pages, and we highly appreciate that the viewers have actively joined our discussion and open forum. Okay, so this year's theme of the National Biotechnology Week celebration is responding to the challenges, business opportunities in biotechnology. The 18th NBW aims to feature local biotechnology products and technologies and promote the latest biotechnology innovations that are ready to be transferred and commercialized. Thus, for this webinar, the OST Picard showcased 3 of its breakthrough R&Ds on biotechnology and their contribution and benefits in relation to the improvement of the agriculture and aquatic sectors. For this afternoon, we have witnessed the presentation of 3 topics from our 3 distinguished speakers. The first one is on the high yield at lower cost with Nutria biofertilizer for sugarcane, followed by the topic on the nanogold biosensor test kit for African swine fever detection, and finally, the topic on the utilization of marine fishery species in the production of protein hydrolysis. So let me briefly discuss or recap the topics one by one. According to our first speaker, Dr. Padilla, the sugarcane industry specifically suffers from poor growth, flooding, or prolonged drought, and high cost of inorganic fertilizer, among many others. Thus, this results to low national average yield of 60 tons sugarcane per hectare. In response, our partner researchers from UPLB Biotech developed a bacterial-based foliar spray biofertilizer which promotes nitrogen fixation and plant growth, implemented in 2012. So this biofertilizer also acts as a supplement to inorganic fertilizer. Some of the benefits shared by Dr. Padilla in the use of Nutria biofertilizers includes the promotion of healthier and greener plants, promotes tillering, and improves cane and sugar yields by up to more than 30%. In 2018, the technology was patented and the product was registered with Fertilizer and Pesticide Authority. Hence, its rollout to ease the farmers' burden of rising prices of chemical fertilizers in the market. I hope the viewers, especially the farmers, were convinced and inspired by the testimonial shared during the presentation of Dr. Padilla. As mentioned, the Fulmite Agricultural Corporation is currently working on its label expansion of Nutrio for rice, corn, and leafy vegetables. So any interested individuals may also contact FAC or visit their website at nutrio-foliar.com. The next topic was an output of the CLSU project, which was
initiated with the proclamation of the nationwide state of calamity due to ASF back in 2021. So this is as per Presidential Proclamation number 1142. There is an urgency to rehabilitate the hog industry of the country to ensure availability and affordability of pork products and subsequently attain food security. In response, CLSU developed the ASF Nanogold Biosensor Rapid Test Kit that pioneers in amplifying the viral genetic material from samples taken either from the animal and from inanimate objects. So the kit uses the combination of lump primers targeting the P72 gene, which is responsible for the viral attachment of the ASF virus, and gold nanoparticles to stain the amplified products. So shown on your screen is the summary of the components of the test kit, which includes the DNA extraction reagent, the premix, and the gold dye for the amplification reaction. So according to Dr. Kla, the amplification reaction takes only 35 minutes as compared to conventional PCR and ELISA. And likewise, the cost of one test is more affordable since the test has its own DNA extraction step as compared to PCR and ELISA, which are requires external commercial extraction and are limited to a number of samples only. The last topic for this afternoon focuses on the utilization of marine fishery species in the production of protein hydrolysates. So as mentioned by Dr. Peralta, the protein hydrolysates are chemically or enzymatically hydrolyzed proteins consisting of varying sizes of peptides with functional and biological activities. So marine fishery species are utilized in the production of protein hydrolysates, including mollusks, fish species, and seaweeds. So with this, the current issue to produce better food products for better survival and maintenance of good health of immunocompromised and general populace is being addressed. To date, a total of 10 chemical and 10 food-grade hydrolysates products were produced by the team, including 1 core blend product powder and 1 blended product powder. During the question and answer forum, very interesting questions were asked to the project team. So to highlight a few, it was asked that for our first speaker, it was asked whether the Nutrio can be used for high-value crops, particularly those from upland farming. Also, may mga na-mention din na different crop commodities, but according to Dr. Padilla, while the Nutrio was developed for sugarcane and that there are no specific tests conducted by the team towards those other products, statements or testimonies from farmers showed that Nutrio was also found beneficial as observed to prolong flowering and high yield. For our next speaker, one of the questions that we may highlight, that we can highlight, focuses on the criteria to ensure fairness in evaluating the test assay. So according to Dr. Domingo, it should be made in an integrated approach and the value should be standardized to remove bias. Also, primer should be designed in the same location of the target gene, among many others. This is to ensure that the tests are reliable and efficient. For our last speaker, one of the question was on the potential use of protein hydrolysates for preventive viral infection. So Dr. Peralta highlighted that definitely there is a potential. How the food products developed by the project contains bioactive compounds that enhance body regulatory functions. And likewise, these hydrolysates produced by the project were tested and quantified for its antioxidant activity, antimicrobial, and anti-hypertension properties. I would like to extend our thanks and congratulate our 3 invited research speakers for the very informative presentations. That includes the synthesis of this webinar and the S&T applications of the biotech applications. I hope all of our viewers learned a lot from today's topics. And once again, thank you so much for joining us today. Back to you, Dan.
Thank you as well for that summary, Ms. Alisa. And we would like to remind everyone who would like to receive an e-certificate from this webinar, you may accomplish our customer satisfaction feedback form, which can be accessed through the link shown on your screen. So make sure that you are registered at the Picard Events website so that you will be able to receive a copy of your e-certificate in a week. Again, make sure that you are registered at the Picard Events website so that you will be able to receive the copy of your e-certificate in a week. And if you are not able to receive your e-certificate, you may notify us at crd@picard.dost.gov.ph. Now, to give us the closing message, here with us is the officer in charge of the Crops Research Division, Dr. Alan Biciano. Dr. Alan?
A pleasant afternoon to everyone. The technologies presented by our resource speakers this afternoon are among the front runners in advancing the application of biotechnology in the field of agriculture and aquatic resources. Evidently, biotechnology has provided contributions and benefits to agriculture, sustainability, and economic stability. The OSTP card continues to bank on biotechnology research to close the gaps in agricultural production and promote sustainable agriculture and consequently provide a greater impact in our society. We are thankful for the hard work, continued support, and unending passion of the project teams in delivering the expected outputs of their research. In particular, we are thankful for the guidance and leadership of the experts from the National Institute of Molecular Biology and Biotechnology, or Biotech, of the University of the Philippines, Los Baños, the Central Luzon State University, and the University of the Philippines, Visayas. Our experts not only gave us a good discussion on the accomplishments of the projects, but also help us better understand and appreciate biotechnology and its application in the agriculture, aquatic, and natural resources sector. Rest assured that DOSTP card will continue to support the responsible use of biotechnology as a tool to address the challenges in the agriculture and aquatic sectors, and subsequently promote awareness of the economic and social benefits of biotechnology. We will be looking forward to more advancement brought by agricultural biotechnology and will support our experts so that together we will have an effective and efficient agriculture sector. I would like to thank you all for attending this webinar. I hope that you found this activity both useful and interesting. Likewise, I hope that you will continue joining the week-long celebration of the 18th National Biotechnology Week. Make sure to like and follow the NBW and DOSTP card page on Facebook to learn more about the exciting activities and webinars about the latest in biotechnology in the country. Again, good afternoon at mabuhay tayong lahat.
Thank you, Dr. Alan, for your closing message. And thank you, everyone, for attending this webinar. And I hope that you will be able to support our other activities here at the OSTP card. And in line with the national policy on biotechnology, the OSTP card supports the promotion of safe and responsible use of modern biotechnology and its products as one of several means to achieve and sustain food security, equitable access to health services, a sustainable and safe environment, and industry development. Again, I am Danica Sembrano, your host from DOST Picard. Thank you and happy National Biotech Week.